The liver performs hundreds of functions and has an enormous capacity for self-repair. It tirelessly protects the body from starvation and defends us from pathogens and toxins. The liver is positioned in the upper right quadrant of the abdomen against the diaphragm, where it is partially protected by the rib cage. The liver is divided into 4 lobes: the right, left, caudate, and quadrate lobes. The falciform ligament separates the left and right lobes anteriorly. Each lobe is further divided into segments that correspond to branches of the hepatic vein. The common hepatic artery supplies oxygenated blood to the liver, but the interesting thing about the vasculature of the liver is not the hepatic vein or the hepatic artery, but the portal vein. While the liver is part of the digestive system and plays a key role in helping to digest fats, the main reason why the liver is so essential for life is that the nutrient-rich venous blood from the entire digestive tract passes directly into the liver through the portal vein. The liver processes the glucose and amino acids in the blood and is responsible for ensuring the body's energy needs, which can fluctuate dramatically between eating and fasting. Portal blood also contains signaling molecules secreted by the pancreas, intestine, and spleen, allowing these organs to communicate directly with the liver. The liver absorbs and stores essential nutrients, including iron, folate, and vitamins A, D, and B12. However, portal circulation also brings with it toxins, such as alcohol and even pathogens and their products. Therefore, the liver functions as one of the body's most important immune organs and contains a standing army of resident immune cells, including Kupffer cells, Pit cells, dendritic cells, and natural killer T cells. How does the liver manage all of these diverse functions so effectively? The answer lies in its architecture and diverse range of cell types. Hepatocytes account for about 78% of the tissue volume of the liver. Cholangiocytes, sinusoidal epithelial cells, Kupffer cells, etc. account for another 6% of the volume. The functions of the liver are made possible by its complex spatial architecture. Branches of the portal vein extend radially to form hexagonal structures called lobules. The liver contains about 1 million of these basic structural units. However, the basic functional unit of the liver is the sinusoid. Venous blood enters the sinusoid through a branch of the portal vein, where it is joined by oxygenated blood from the hepatic artery. The sinusoid is lined by a row of sinusoidal epithelial cells that form a barrier porous to small particles, while intravascular macrophages, called Kupffer cells, ingest larger particles. We pass through a fenestration in the epithelial cells into the parasinusoidal space, also called the space of Dis, where we encounter a dense row of 15 to 25 hepatocytes. Hepatocytes are the cuboidal epithelial cells that perform most of the liver's essential functions. Hepatocytes contain a large number of mitochondria and may be polyploid or even binucleate. Hepatocytes synthesize multiple proteins, including serum albumin, fibrinogen, and several clotting factors. The liver is one of the primary targets for insulin, where it stimulates the conversion of glucose to glycogen in the process of glycogenesis. Conversely, glucagon secreted by the pancreas stimulates synthesis of carbohydrates via gluconeogenesis and release of glucose into the plasma through glycogenolysis. Unlike other nutrients, lipids do not enter the portal vein, but instead pass from the intestines into the lymph vessels. However, the liver retrieves lipids from systemic circulation and synthesizes cholesterol and bile acids. This ability to interconvert biomolecules between one form and another is essential to the liver's role in maintaining homeostasis. This is not only important for metabolism, but also essential for detoxification. Ammonia is a byproduct of protein digestion that is toxic to the central nervous system. The liver converts ammonia to a water-soluble form, urea, that can be excreted in the kidneys. The liver is also responsible for clearing bilirubin, a byproduct of erythrocyte turnover. Accumulation of bilirubin in the skin and eyes causes jaundice and may be the first indication of liver disease. As the first line of defense against xenobiotics, the liver also plays a critical role in detoxification of ingested substances, using a series of reactions involving enzymes such as cytochrome P450 oxidases, glutathione S transferases, and efflux transporters. Many prodrugs are designed to require detoxification in the liver in order to become active. A hepatocyte normally lives for about 5 months, but a damaged liver has enormous regenerative potential. Mature hepatocytes have the unusual ability to re-enter the cell cycle. Following surgical resection of part of the liver, hepatocytes can rapidly divide and restore the lost tissue volume in a matter of weeks. Hepatocytes are 1 cell layer thick and attached to neighboring cells through tight junctions. This junction also contains a small space called the bile canaliculi, into which hepatocytes secrete bile. The canaliculi drain into bile ductules formed by cholangiocytes, and then into bile ducts and eventually the common bile duct, which joins the pancreatic duct and empties into the small intestine through the ampulla of Vater. Most of the bile salts are reabsorbed in the intestine and returned to the liver through portal circulation. The space of Dis also contains hepatic stellate cells, or Ito cells, which store vitamin A. However, when stimulated by Kupffer cells, these cells promote the development of liver fibrosis through excessive deposition of extracellular matrix proteins, leading to progressive loss of liver function. Within the space of Dis, portal blood passes down a gradient across 3 functionally distinct zones. Zone 1 receives oxygen and nutrient-rich blood and is specialized for oxidative functions, such as gluconeogenesis, beta-oxidation, and cholesterol synthesis. Zone 3 contains a high concentration of cytochrome P450 and performs non-oxidative functions such as glycolysis, lipogenesis, and detoxification of xenobiotic compounds. Blood passes through zones 1, 2, and 3, and then finally empties into the central vein, where it returns to systemic circulation through the inferior vena cava. The liver is a remarkably adaptable organ that can metabolize and detoxify a wide range of substances. However, because the liver encounters such a diverse range of foreign substances, it must be relatively immunotolerant to avoid triggering a nearly constant state of inflammation. In the next video, I'll talk about how 2 viruses exploit this aspect of the liver to establish a chronic infection that can ultimately lead to cirrhosis, hepatocellular carcinoma, and death.